All right, everybody. So our next speaker is Professor Ian Lane, who probably needs no introduction here in New Zealand. So he is a leading authority in medicine, nutrition and management of dairy cattle. He is a graduate of the University of Sydney, and he spent 6 years as a large animal veterinarian in the Hunter Valley before undertaking his PhD at the University of California. He actively consults to dairy and beef farms across Australia and has extensive New Zealand experience. And in fact, I heard his name mentioned on farm in the Potato area just last week. So he's lectured in clinical nutrition and cattle medicine at the University of California and then the University of Sydney, where he remains an adjunct professor in veterinary clinical studies. Extensively published and as the author of more than 200 scientific articles. So very excited to have Professor Ian Lane here, and he's speaking today on a review of acidosis and thinking beyond pH, which I think will be a nice follow-on from Professor Greg's presentation this morning. So we've got a couple of post-it notes starting to build over here with questions. Please do keep writing them on those post-it notes for something for our panel discussion, or if you're a little bit shy to shout out, then feel free to use the post-it note system. And as we've mentioned, yes, certainly if you've got a quick question on a slide that's relevant, feel free to shout it out. Our webinar people, I know you're having a bit of a hard time hearing the questions from the crowd, so our guest speaker will repeat the question for you so you can hear that too. So yeah, welcome to the stage, Professor Ian Lane.
Thanks, Nadine. Is that working? Can you guys hear me? Yeah, look, it's a real pleasure to be back in New Zealand. It's always a joy. And I just, really appreciated Greg Penner's talk. It's a terrific talk. But one of the things that came up in just discussion post that was a little bit of a history story. And a number of years ago, a good mentor of mine, we were talking about transition diets. And he said, of course, you know where the story of steaming up came from. And I said, no, tell me about the history. He said, well, there was a rather pompous little British professor by the name of Bobby Booflower. And Bobby Booflower went around the world talking about steaming up because in the 1960s he had a 10,000 litre herd, as in a bona fide, all the cows averaging 10,000 litres. And the thing that he'd done was he steamed these cows up, and that was what he was going around the world telling people about. And of course he steamed them up with barley, but my old friend said, by the way, Ian, the barley wasn't barley. I said, well, what was it? He said, it was brewer's grain. And there you are, completely different. Low starch, low DCAD, in fact a negative DCAD, and a completely different concept, high in good fats. So, it really is all about ration balance and not getting too caught up in things. Oh, I really want to pay acknowledgement to my good colleague Helen Golder. We work together very closely. She's our research director at Scibus, and a lot of the information is in our collaborative work. And like Greg, much of this depends on PhD students over time, people like Liz Bramley. So you Kiwis are a cultured lot, so we're going to start with a little Shakespeare. What's in a name? Romeo asked. He said, thou, a rose by any other name would smell just as sweet as thou, sweet Montague. Of course, the name thing was a bit of a problem for Romeo and Juliet, as you might recall. So it's a really important aspect to get some of the definitional issues correct. And when we start talking about a name, is there a problem? Well, yes. If you're using the wrong nomenclature and you're getting the wrong messages, you end up with wrong results. And a great example was really the years we spent trying to acidify urine instead of trying to prevent milk fever. So we went down completely the wrong path, we got the wrong understandings, and we have the same problem with acidosis. So I am going to refer to ruminal subclinical acidosis or clinical acidosis. I'm not going to talk about subacute. Why? Because subacute has no implication of production loss or health impact. So you've got to think about, what does a subclinical disease also deliver to you? Is a subclinical disease something that is going to have an impact? Okay, and what sort of impact does a subclinical disease have? If you think about ketosis, it doesn't have, as a subclinical form, profound impacts that are immediately obvious. They're fairly subtle. There are risk factors for decreased reproduction. There's risk factors associated with decreased health. So you need to be careful when you start to think about these things. They're not always profoundly obvious. Is acidosis a problem? I don't have Kiwi data that's analogous, but in Australia, with our study, which was 800 cows in 100 herds from Liz Bramley, what we found was a prevalence of 10% of lactating cows in their first 100 days in milk. That's a lot of cows. We'll come into some other further thoughts. Our recent study, in much more limited numbers in Australia, was a 37% prevalence of acidosis. So if we consider that a case lasts about 2 days, and some of Greg's data clearly showed that sort of link around that limited period of time of an impact. If you think about a case lasting 2 days, roughly in a prevalence of 10%, and if you think about that whole lactation data, and again, some of Greg's data showed that it extended over the whole lactation, that would amount to about 15 cases per cow, or 1,500 cases over a 300-day lactation. So if that is associated with production loss, you can start to think about just how much loss might be occurring in herds with a high prevalence of acidosis. So what do you expect from a clinical disorder? A subclinical disorder, you do not expect profound effects, you expect relatively subtle effects. Otherwise, it's a clinical problem, isn't it? Okay, so you've got two categories, really three, I suppose. One, nothing. Two, subclinical. Three, clinical. Okay, so they're different conditions. So definitions of disease often have reflected historical generation, and the great one is milk fever. Do you find fever with cows with milk fever? No, most of them are cold, aren't they? In fact, if they do get down with milk fever in the hot sun at home, well, you can cook them. But basically, it's not a function of those animals having a fever per se. So, we need to think about using strictly defined terms, and we need to go back to some postulates. So I modified a few years ago Evans' postulates for metabolic disease. And that way, we were able to think about how we structure and understand the disease. So current definitions just aren't fit for purpose. Greg showed a number of different cut points, some with areas under the curve, time of day, you know, different points of inflection on pH. And Gary asked the question, well, which pH are we talking about? Are we talking about the one near the apical villi, or are we talking about one somewhere else? Okay, so the question is, are those fit for purpose? And I don't think so. Okay, so as I said, we modified Evans' postulates. We had a look at what we might be able to do by modifying those postulates for metabolic disease. And I'm not going to go through all these, but there's a series of tests you can apply to this condition to find out whether it makes sense or not and what your definition truly is. And if we look down, you know, there's a number of measures here that are important. We discussed these ones. These ones are a little too hard to discuss in the environment today. But if you have a look now at some of the research we've done and others have done, we're going to go through these postulates a little bit and look at what the responses might be. So one of the first things that we want to look at is to demonstrate in prospective studies that the exposure to a putative cause increases the disorder. Now, what I'm going to talk a little bit about is some eigenvectors, and I'm going to explain those in the next slide. But all I need to tell you is that if you've got an eigenvalue of 1, you've got subclinical acidosis. If you've got an eigenvalue near 0, you're pretty safe and you don't have it. But in this study, what we did was we fed supplement, and that supplement was about 80% wheat. And so we fed it either as a slug in a milking parlour environment, or we fed it as a PMR, partial mixed ration. And what you can see is that the animals that got 16 kgs and it was slug fed spent most of the day in an acidotic state. You can see that at 14 kg, again, most of them. You can see 12 spiking, and you can see that at 10 and 8, there was relatively little risk. And if you look across at the PMR, by delivering the feed as a mix with forage, you reduce the risk. So one of the messages is, yes, we do have prospective studies showing that the exposure increases. Experimental reproduction of the disease should occur within greater frequency in cows exposed, yes. A spectrum of responses from mild to severe along a logical biological gradient, that occurs. And measurement of change in metabolism should result from exposure to a putative cause, and we can prevent this condition by exposure to decreased or controlled measures to eliminate the disorder. And all those biological relationships should make epidemiological and biological sense. So those are the criteria we're looking at. So instead of just looking at pH, we looked at much more of the ruminal environment. I think this is a big step forward in understanding acidosis. I think some of the stuff that Greg talked about in particular is going to advance it even further as we start getting into these biomarkers and we start looking at measures of damage to the rumen as well. But if we look at this Bramley model, what we have is rumen ammonia, lactic acid, pH. We've got 7 individual volatile fatty acids measured, and then what we looked at were the outcomes. So we looked at health data, and we were able to demonstrate that the acidosis as we defined it using these mix of measures was associated with a higher prevalence of lameness in herds. So herds that had a high prevalence of acidotic animals also had a much higher prevalence of lameness. We were able to look at the production data, and we'll show you some of that, but there was an impact on production, and we were able to develop that acidosis score, but we were also able to associate diets on that 100 herds back to risk of acidosis, and the logical exposures were there. So low NDF, high starches, high NFCs, non-fibre carbohydrates. So just looking again at these sort of measures, we used a discriminant analysis model. I don't think that'll interest you too much. And then what it did was allow us to produce an eigenvalue for each animal so that we could determine where it lay in terms of a risk profile. So, was there an impact? There's got to be an impact if it's a subclinical disorder, okay? And yes, there is. As you go up, the milk yield started to go down as you hit 14 to 16 kgs of supplement. And you can see the milk fat percentage started to fall as you hit 14 to 16 kgs of supplement. Makes absolute sense that you've got that problem. What else happens in terms of removing risk? Well, if you use potent agents to remove risk, such as virginiamycin, and another one was buffers, what you can see is that you've got a reduction in risk of cyclic eating. And from this data, we could see that it's about a 2-day duration of cyclic depression in appetite before the animals start to recover. And one of the things to contemplate in our data, and one of the things we struggled to understand when we first did Liz's study, is that if there's about 10% of animals that are undergoing a subclinical acidosis, there's probably a whole bunch of animals in there that are recovering from subclinical acidosis. So we had 3 distinctive groups. We had one group that was obviously acidotic, we had one group that was obviously not acidotic, and we had one group we didn't know actually what was going on. And so it's taken us a long time to kind of figure out that other group. So cyclic eating bouts last approximately 2 days, and that demonstrates that the disorder should follow exposure to the putative cause, and removal or modification of the exposure should reduce the disorder. So what is ruminal acidosis? It's a complex of conditions. I think of it as a continuum, okay? So it's a sliding scale of risk that occurs as animals are exposed to a rapid increase or change of rapidly fermentable carbohydrates when there's insufficient physically effective fibre. And this becomes a big issue for New Zealand pastures. I've just been looking at some pasture results from, farms that are pretty local to here. And what you're looking at now is NDFs around 28 to 32, and you're looking at proteins that are around 35, 36. This is last month. And so what you're talking about is that fibre is just not physically effective. And many of you know my good friend and former student, Charlotte Westwood, and, you know, we've often looked at, you know, that strength of comminution of pasture. So can you pick that pasture up and just pull it apart? There's a break in your hands easily. And if it does, it's not going to deliver much physically effective fibre, is it? So you've got to think about the physical form of the diet as you think about it. So that leads to an accumulation in the rumen of organic acids that reduce microbial protein yield and fibre digestion. So those are the problems, and it results, as Greg so elegantly showed, in a decrease in dry matter intake, and of course that decrease in dry matter intake is a major problem. So causes and types of acidosis. You've got strong acid formation, and the strong acid is lactic acid. Lactic acidosis is part of the continuum, but it's the extreme end. When you get a lactic acidosis, that's when you've got a clinical acidosis. If you look at the other forms, we've got the most common form in dairy cattle, which is increased weak acid production, so volatile fatty acids. You can also get into trouble with an acid dump. Is there much eelage around in New Zealand at all, any corn eelage? Well, yeah, I've never seen any. Thanks, Sue. Bottom line is that if you do corn eelage, you run a reasonable risk of dumping lactic acid in big mobs into the rumen, and it will function in a disruptive sense. Removal of agents that stop acid formation. So if you, and we've seen that situation back in Australia, especially when there was more Virginia mycin removed, if somebody left the VM out of the diet, well, you would see those cows break with acidosis. So if you took away the controlling agents, you'd have a problem. Now, this is where the area gets interesting. There's also the impact of vasoactive agents. So again, Greg's referred a lot to the lipopolysaccharide, but it's not the only vasoactive agent. In fact, lactic acid itself is an agent that will cause vasoactive impacts. You've also got histamine. You've also got insulin. In the equine, insulin's associated greatly with laminitic change. So those are some of the things. And lastly, don't forget the foregut and the hindgut acidosis. So it's not just hindgut, it's foregut. And the sequelae, well, what are the sequelae? We do see quite high rates of liver abscessation, especially in Holstein cattle. So cows and steers, Holsteins, very susceptible. So differentiate the one. You've got acidosis here and lactic acidosis up the top. And when I think about it, and we really have the perfect first talk, because Greg focused on his tremendous work showing the clearance through the ruminal epithelium and the clearance of acid out of the rumen, but I want to talk about the generation. So the two talks match up perfectly. The severity is based on the animal's ability to safely sequester hydrogen. So in other words, those volatile fatty acid absorption, taking it out of the rumen, putting it across the rumen wall, that gets rid of the hydrogen risk. It's an anionic risk. Okay, so it's about the speed and the quantum of hydrogen creation, because if it's too damn fast, the rumen can't clear it fast enough. So what we've got is the rumen, and if you take starch, here's a safe place to sequester starch. It goes into protozoa. They sequester starch and the slow release is safe. You've also got bacterial glycogen, and that's another safe place to hide the hydrogen. Okay, so sequesters it and safe. And you've got starch, and if you've got rumen degradable protein, in other words, you've got ammonia there present, and amino acids and peptides, then you can create microbial protein, which is a fantastic safe source to put hydrogen in. There's about 56, I think, grams of hydrogen per 100 grams of bacterial protein. And of course, methane's also produced, and that's a safe sink. If we look again at the pathways around hydrogen, hiding hydrogen, starch breaks down to glucose, glucose goes through the glycolytic pathway to pyruvate, and then it goes to those safe or relatively safe volatile fatty acids. Alternately, it can go via the succinate pathway to propionate. It also can go down from pyruvate to lactic acid, and that's the one we don't particularly want. It's unsafe, and it's often transferred off to propionate, but it's also transferred off to valerate. And valerate is a very, very sensitive indicator, as is propionate, of risk of subclinical acidosis. So again, if you now take sugars, something like sugar beets 83%, NFC, so you take those beets, and what you can then get is glucose, and you get pyruvate, but then you can get lactic acid. So sugars are profoundly different to starches in terms of their risk, and then again, you've got all those different pathways that are available, but the speed of breakdown of sugars is really great. And if you get rapid breakdown, then it's harder to transfer all these processes. They all take time. So hiding it in the rumen, sugar's difficult. And of course, we've talked about saliva. I think I'm going to have to put in a new series of slides to complement Greg's work and put in the clearance in the future. So acidosis diagnosis, why not pH alone? Well, Gary's already asked the question, and you know now why not pH alone. But all these sites are different. Anterior reticulum is different to the rumen mat, is different to the dorsal part of the mat, is different to the bits near the wall. So what pH are we talking about? Okay, it's not homogeneous. pH of the rumen is only one factor that causes pathology, and it may only be, well, when you think about subclinical acidosis, there's no real evidence that I'm aware of that you're going to have an impact that's going to erode the rumen, okay? So you've got to be careful about that. pH varies time among cows, and again, some of Greg's evidence showed that, but we got some nice evidence from my friend Nick Johnson, who I've just blitzed. Ooh, can we get that back up? Jumped out of that. Sorry about that. Yeah, I've killed it. Totally trashed it. So Nick Johnson's work, these are indwelling probes in the UK, and what Nick's work shows is that there's basically a lot of variation in individual cows. And so when you're talking about this variation around the individual cows, it's both within and between, within a day, and it's also between cows. And again, some of Nick's work's just showing just how profound the differences in pH are during a day. Some of these cows sitting below 4. Some of the cows in the study with Liz Bramley's were running around perfectly happy with pHs of 4.5. Now, when I read the textbooks at that time, at a pH of 4.5, that cow was supposed to be dead. So, I don't know why she was looking happy and healthy, but the point really being that is the sort of extreme that you see. And of course, those same cows, one with a pH below 4 is running around with a pH of 8 at a different time of the day. So you've got to understand why pH is such a poor measure. So, feeding and type of substrate, this is pretty clear. If you're talking about 1.2% of grain in a challenge study, those cows are running around with a rumen sample taken through a stomach tube, and our stomach tube gets down into the rumen, okay, doesn't just sit at the cardia. They've got a rumen pH of around 6.8, say, at the end of the time. But if you feed them grain plus 0.4%, so again, 1.2% of body weight, and you give it to them as fructose, it's a completely different rumen pH that you result in.
Sorry, again, what type of grain was that?
Yeah, those were lactating.
What grain was that?
Oh, grain? I think that one was wheat, yeah. So, if we look at how many of you are familiar with receiver-operator curves? Anybody? Nobody. Good, excellent. Let me just explain this. On this one axis, you've got the false positives, which is 1 minus the specificity of a test. On this one, you've got the true positives, the sensitivity. So you want a test to be both sensitive and specific, right? Which means that the better the test, the closer it is to 1 in this corner. So let's look at some tests. This is valerate and propionate diagnosing subclinical acidosis. They're really close, they're above 0.9, so they're bloody good. If you look at, for example, ruminocentesis or rumen pH with a stomach tube, they're almost identical. and they're around 0.6 odd. Okay, so they're not a bad test. Don't throw them away. Don't ignore them totally, but just understand that you're going to get a lot of false positives and false negatives compared to using valerate or propionate. And, you know, the Europeans are very keen on butyrate sometimes. I'd have to say that that's running like chance. Okay, so this middle line, if it's close to chance, it runs along the middle line, and you can see both butyrate and acetate are not good tests for subclinical acidosis. At the moment, unfortunately, the volatile fatty acids are pretty much not a cow-side test. And we think there'll be cow-side tests for this much better. You know, is there a great similarity between a stomach tube pH and a ruminocentesis pH done at the same time? This is from Liz's study, and you can see that the R squared is not high. So you're not necessarily getting the same thing if you're doing a stomach tube pH versus a ruminocentesis. So what about indwelling probes? Look, the indwelling probes have a lot of promise. I think they're a really exciting technology, but there have been problems with the rumen probes, and you know that the one in New Zealand sort of disappeared. Fistulates are not physiological, and you're down to limited numbers, and both are limited in capacity to relate to larger populations, although there was an excellent paper just out looking at indwelling probes for robotically milked cows, and that was a really quite valuable paper. So, assessing rumen function, how are you going to diagnose this in the field? Yeah, Kurt.
But you had the protein ratio there too.
Yeah.
Which is a very easy one to do.
It is actually, and it's a good one, and with your interests, that's highly appropriate. It's not a bad test. It's sensitive but very non-specific. So, you know, that's the problem. It'll pick it, you know, all the herds is, I don't think I'm showing Liz's data today, but there was a marked reduction in fat to protein ratio with the acidotic herds. And so, you know, yeah, it is a really good indicator to ask the question. So, yeah. Yeah, Gary. Whoa.
Just intrigued about your hydrogen in your rumen and tests and stuff. Given the fact that methane is a sort of quite a good hydrogen sink,
Yeah.
do you think a low methane animal would be more prone to acidosis?
I think there's a distinct possibility. You've got to hide the hydrogen somewhere. And, you know, when we're thinking about these, you know, these grand strategies for, you know, reducing, yeah. I mean, a good example is monensin, you know, and I know you'll work on monensin, so we'll leave that aside for a second. But if you think about the monensin, yeah, monensin decreases methane production. Where does it put it? Into propionate. And I'm not quite sure where some of the interventions now put the hydrogen. Okay, so, you know, I'm quite concerned about the fact that we're going into some of these things without potentially thinking through exactly the issues you're talking about. I think so. You can see how much mayhem you can cause unimpeded. I love it. So if we're looking in the field, how are you going to, oh, BJ.
You looked at rumination times?
Oh, gosh. No, I haven't. Yeah. It's a really good comment, and you might be able to pick up the acidotic cows with a decrease in rumination. So I think that's something else that could be certainly looked at in terms of monitoring. And we haven't been there yet, so I reckon that's a really, really good comment. A whole lot of work to do. So in the field, how are you going to know whether you've got an acidotic situation or not? And I think it's, you know, first clinical signs of disease. Are you seeing lame cows? Are those lame cows associated with a laminitis? Are you seeing, you know, animals that are basically going off feed, respiratory disease? You know, are your cows, when they go to the slaughterhouse, have a high incidence, have a high prevalence of liver abscess? So those things are important. You've got the disease and culling history. You know, have you got epistaxis, the bleeding out the nose? Look at the feed management practices, the ration. Is the ration well formulated? Look at the rumination percentage and rumen fill. Are the cows actually ruminating when they're lying down, or better still, maybe you can measure it with collars. Fecal exam, are you looking at the splashy, bubbly faeces that are associated with acidosis? Milk fat and protein ratios. So yes, it's part of your investigation, and the rumen measures. So you've got a whole lot of tools that you can use to rely on in the field to help you decide whether you're dealing with an acidosis. And if you look at your risk, of course, you know, your low risk and good rumen function, high NDF, low starch, low sugars, healthy tissue and stable diet. Obviously, you've got to balance that sort of ration with lush pastures, and how do you manage to get your production levels up while still controlling your risk of acidosis. So we're always going to be sitting a little bit near the precipice because we want to optimise rumen function at the same time as we don't want to put our animals at undue risk. So optimising rumen function, offer a balanced ration. Avoid rapid feed changes by adapting cattle to new feeds. Minimise slug feeding, feed some grain in a mixed ration. You know, a lot of New Zealand farms have feed pads, and it's a sensible place to feed rather than in the parlour because you can integrate the forage and the grain together. And as you saw with our data previously, using a PMR versus a slug-fed diet resulted in a marked reduction in acidosis risk. So it's a logical place to be. Feed at regular intervals, more frequent feeding's preferable. Use a transition ration 21 days before calving. But you don't have to steam them up with heaps and heaps of starch, okay? You've still got to meet their protein and energy goals. And one of the things that can get lost in these discussions is a need to keep those animals in a positive nutrient balance. They need to have a positive protein, mineral, vitamin, and energy balance, okay? I always talk about nutrient balance. I try to avoid energy balance because it just focuses on one part of the pathway. So, feeding rumen modifiers, check if they're in at the right dose. You know, there's a real tendency to put rumen modifiers in at the wrong dose. Monitor cattle and performance. Check the faeces, rumen fill. You know, I just saw a herd last week which, you know, really did have some problems, actually the week before. And it had classical acidosis. And when I looked at those cows, they had a rumen score of about 1 to 1.5. They were all flat. Determine the feed value of all feed components, feed test. You know, don't assume pastures are just pastures because they ain't, okay? It's like oils ain't oils. You've got to think about your pastures and what they're delivering. As you know, the Italians are really different to your perennials, and your perennials can vary at time of day, as do your Italians. So you've got to think about what pastures am I actually feeding. I think I've often said in New Zealand that if you went to, you know, BHP and you said to BHP, you know, tell me about iron ore, and they'd say, well, it's brown and lumpy and lives in big mountains in Western Australia, it wouldn't be good. But if you go to most Kiwi farmers and you say, tell me about your biggest single input to your cows, well, it's green and it grows, you know? So you've got to quantify things. You've got to take it a little bit further. Ensure all cows, especially heifers, have adequate feed. Don't overmix. Aim for a chop length of about 5 centimetres. And then you can find that, in fact, there's an interaction between diet and what bugs are in the rumen. So we're very interested in the bugs in the rumen. And what you can find is that in a single pulse challenge, in this case, grain histidine, grain fructose and histidine, grain and fructose, what we found was that in a single pulse challenge, which was just suddenly slamming these guys with a fair bit of extra grain or grain and fructose, we immediately didn't get much change. But in a longer term study, and that's the PMR plus protein and the controls, what you can see is that with time you get an adaptation and you find that the groups become a lot more different as they adapt. So clearly once you adapt cattle, they develop a different microbiota or microbiome, and therefore they're more capable of responding. So we're going to look at now optimising rumen function and some new findings, because we've got increased interest in exploring the host microbe interactions and the phenotype microbe interactions. And what we're doing is using these omics methods and gene-wide association testing. So it's a little bit esoteric, but it's really important because now we can start to look at the relationship, and this wasn't our work, this is from Zoo, but what you can see is you can look at the outputs, fat, protein, milk yield, lactose. You can look at the volatile fatty acids in between, and then you can start to look at the associations of the bacteria with those. So now we're starting to understand the rumen at a much deeper level. Okay, gene-wide association studies. Here's one of ours. We're looking at those VFAs, pH, bacterial abundance in family and in phyla. I'm just going to show the phyla results to keep it simple. Ammonia, lactic acid, and then we had our acidosis eigenvectors. So we categorised the cows. So this was our first study, and it's a very tiny study. It's only about 40 heifers, but what we had was a pre-adaptation period where we just brought them in, taught them how to eat. Then we had 10 days where we adapted them to a TMR plus feed additives. Then we had 10 days where we put the feed additives in and 0.1% fructose. And then we finally challenged them with 1% wheat plus 0.2% fructose and the feed additives. And we had an NFC of 76. Now that might seem incredibly extreme, but just think about those cows eating fodder beets down in Southland in the winter when all the green stuff's gone on top. They will probably be around the same as this. Okay, so what did we find? We measured those things I've just told you about. What we found was this incredible variation between animals in response. And for the first time in our studies, because we'd rarely found lactic acid, we were probably creating ruminal lactic acidosis. And you can see some very high total lactate measures in this study. But look at the total lactates. It's had some cows breaking out and going berserk with really high ruminal lactates, and you had some of these heifers virtually untouched, virtually no lactate. So we're looking at this same animal, very similar animals. They're all off the same farm, probably out of the same sire, and what you've got is huge variation. So the acetates, the rumen pHs, and the rumen valerates. And when we got these data, we thought, you know what? Why the variation? And possibly this is down to the mammalian genome. So, to the best of my knowledge, these are amongst the first studies to look at rumen function versus the bugs versus the mammalian genome. And it's the first stuff in any species that looks at the production aspects as well as the interaction between the host, the mammal, and the gut. And as you all know, we are actually compositional, constructs. We're not just mammal, we're mammal plus bug too. Okay, so when you think about it, it's really important to understand that. So what did we find? In short, we found these QTLs, which is gene markers. Basically, we found them for a whole bunch of things like acetate to propionate. We found them for lactate, and importantly, we found them for eigenvalues. And what we were able to also see is things like chlorflexi, phyla, the firmicutes, which are very important, and the tenericutes, which seem to be important for our acidosis. We found that there's genetic markers for those. So, conclusions, genetic associations occurred, and within the ruminant environment and acidosis risk, we needed to look at associations in a larger population. So what do? We got with our buddies in different parts of the world, up in Canada, California and Wisconsin, and we ended up with 36 farms and about 320 critters in this study. And so we had 8 pasture, 1 TMR, 1 PMR in Aussie, and the rest were TMR farms. So we took information from the cow, blood or hair for its genes. We took the diets, got information on them. We took the rumen, those things we've talked about, and then we looked at the milk outcomes as well. So what did we find? We found pretty high risk of acidosis. So subclinical acidosis, again, it reinforces that it is a problem in herds. We found the high-risk group had rumen phyla fermentation production characteristics consistent with the model of acidosis that reflected rapid rate of carbohydrate fermentation. Medium risk, they had cows that had rumen fermentation profiles that suggest they may be inappetent, which is very consistent with Greg's work today. So you can see that the tie-ins between the different presentations are there. And they might have not have eaten recently or were in recovery from acidosis. And then we've got a low-risk group. So let's have a look at some of the things that are different. So if we have a look at pH, there's not a lot of difference between the group that was low risk or in the group that was at high risk. But the medium group, if we look at them, they've got a higher rumen pH. If we look across the total VFAs, not much difference between the low risk and the high risk. And again, the medium group, again, are different. They've got low volatile fatty acid concentrations. If we come across and look at the acetate to propionate ratio, again, that tells us exactly what's going on. The propionate is up, the acetate is down. And if we have a look at our friend's valerate, who really does tell us a lot about what's going on, and we look at the other isobuterates, isovalerates, the key thing is to look at the valerate, and the valerate's very different. And we can see that difference occurring quite markedly across the groups. And you can see also that the propionate is elevated in those groups as well. So we've got a greater synergy of energy and protein and a more stable rumen fermentation. And I particularly want to point out the ammonia. The ammonia is very important in that difference between groups. So, looking at the milk measures, what did we see in terms of the milk measures? Well, we didn't see a lot of difference, but you can see that you've got the high group, which was high, and you've got a drop off to the low risk group, and those are trending exactly the right way as you'd expect. And again, with our low risk group, you can see the fat difference to the high risk group. And the intermediate group is intermediate. So, if we look now at regional risk of acidosis, we've got a much stronger risk in Australia and California than Canada. Obviously, they've been listening to Greg and they've got it right. So that's what's going on. But yeah, you can see the distribution and the low risk very much more prevalent in Canada, and you might ask why. Well, here's the Australian rations. So you've got NFCs, about 20% starch. NFCs are modest. NDFs, pretty high. CP's about 19. So you'd go, why were the Australian cows at risk? Well, they're being slug fed. By and large, you've got a lot of slug feeding. So they're not getting the PMRs. If you look at California, no surprise that California's got a problem. Look at the NFCs, they're nearly 43%. Wisconsin, 42%. And the Canadians, being nice conservative members of the British Empire like they are, the Canadians are back at 38 and the sugars are low. Look at the sugar differences. California, up around 7. Australia, 6.3. You go down to Wisconsin, sugars are low. So you're seeing quite a difference in the way the diets are constructed between the regions. Those differences are being reflected in the acidosis risk. And the really, really interesting thing for me is that when we get into the acidosis, we start losing bacterial diversity. So there's a really important concept about a core microbiome that basically keeps the rumen functioning. Then you've got these peripheral aspects of the rumen function and different phyla and families and species that are peripheral to that. But one of the really interesting things to ponder is why would you put up with a parasite like protozoa? You know, protozoa reduces your ruminal efficiency markedly. So why would cattle tolerate that? It's probably because while for the individual you can lose that efficiency, as a population retaining something that protects you under a dramatic challenge like starch is really important to retain for group survival. So we've got to think about this in an evolutionary development and herd context. And what you can see is that the high-risk groups have much lower diversity, and you can see that the medium groups approach the low groups. It's really interesting to look at that difference. So as we change that rumen with a real challenge, you get very different aspects around it. And then you can also look at the dissimilarity of the groups, and you can see the low and the medium group tend to be more similar. The high group is different. And when you look at this, it's only explaining about 5.3% of the variance. So there's a lot of other factors going on, but it's consistent with a subclinical disorder. A subclinical disorder is not profound. Okay, it's important, but it's only going to explain part of your production differences. I'm sometimes surprised how much evidence we have for the negative effects of subclinical acidosis coming out of our studies. It's really important. Okay, region, really different by region. And then when we started to look at the phyla that predicted acidosis risk or associated with acidosis risk, unsurprisingly, it's the Firmicutes. These are the guys that multiply when there's a high starch or high sugar environment. Okay, the Spirochetes, they're sort of sitting up there as being increased. You've got the Lentosphaeria and the Planctomyces, and they go down, and the Tenericutes, which we've found in all our studies, are reduced. If we go down and we look at the low-risk group, again, the really nice thing about this is that the low-risk group has a completely different profile that almost mirror images the other. So if you look at who's elevated in the top, it's the ones that are decreased in the bottom. So what you've got is that split, and it's really nicely executed as you look at the differences between these groups. And the area under the curve, how much of the variation did we explain? A lot. So those two models are explaining about 93% of the variance in that description of whether they're acidotic or not acidotic. So really quite profound tests. Okay, if we take it down to the next level, this I get terribly excited by, because this now tells us what parts of diet are really playing and are really influencing the bacterial populations. And if we're going to improve things, well, this is where we've got to look. And amazingly, we had 78 different phyla that were predicted by, 78% of the phyla that we looked at that were predicted by sugars. So the sugars were really a powerful agent that determined which phyla were present or absent. And similarly, the crude protein also had a very high percentage of those present or absent. So if we look at a graph showing which factors are important, so just to walk you through this graph, the bigger the arrow, the more directly it points to the region, the more important it is. And the arrows that go in the opposite direction to it are also important. So if we look at the California cows, you know they're characterised by quite a strong lactate, quite a strong butyrate, ammonia, and then the species are also associated with California down here. If we look at the cows at high risk of acidosis, you can see the valerate, propionate and caproate. They're the ones that are there. And unsurprisingly, here's the Firmicutes and actinobacteria. So we're seeing a consistency around the expectation of the bacteria that should change with the outcomes of volatile fatty acids, with the risk of acidosis. So it's tying together. So Australia had a better protein percentage and protein kgs as a result of that, and the Bacteroidetes were associated. If we look at Canada, the Lentosphaeria were important in describing it, and it had the isovalerate, isobute, and it had fat percentage increase in Canada. So again, those diets between regions, the outcomes between regions, they are associated with our categorisation of these conditions. So markers. Typically when the geneticists do a study, they look at thousands and thousands of cows. So it would have been a bit difficult to get thousands and thousands of cows. I don't think my collaborators around the place would have really enjoyed having to sample the number of cows that we were thinking about. And more credit to Liz Bramley, who sampled that 800 cows, okay? But if we come across and we look at the milk, you know, we got one marker for protein percentage. Well, that's great. You know, we know there's markers for protein percentage. We got a tendency for acetate, a tendency for butyrate and isovalerate, and we also got a tendency distance to the centre of the low acidosis group. So on a sample size of 300 odd, that's a damn good result. If we look at the markers also for the bacteria, we did get gene markers for a number of the bacteria, and we can come down to the family level, and some of the Firmicutes were there in reasonable numbers. So we are able to find genetic links between the mammal and the presence of the bacteria in the rumen. which is really important because it does suggest that over time we may be able to select for better rumen function. Yeah.
Is there like an adaptation period for each cow? Because I'm looking at all of these countries and I'm thinking surely each cow or each group of cows within the country have adapted for a long period of time to have either higher fight fila or lower or higher rumen or higher body rate or whatever it is. So you had a control against each farm to see what was the delta difference within each farm?
So we can look at that, and I think we did look at it, but I can't recall exactly what we found now. But the variation within farm I would expect to be similar to the variation that we were achieving when we started to split those animals out under a challenge study. Probably less profound, but it'll still be there. I mean, your comment's interesting because the weekend before I was away for a week, I went and pulled out a bunch of old slides, and it was my first visit to America in '83, and so many of the slides were of bad feet. And I just went around photographing laminitic rings, overgrown hooves, and photo after photo after photo of bad feet. You don't see that now. Now, that may be partly because there's much more emphasis on looking after feet, but the other factor, I suspect, is a long-term removal of animals that just don't cope. You know, if it goes lame with a laminitic change quickly, guess what? It's not going to be there to breed more replacements, is it? So I think there's been a strong selection pressure, and that's probably reflected in some of the differences.
So given that in your experience, how much weight can you put in those markers if you're comparing countries because they can fit very differently?
Well, we can go down and break it down, but then we're not going to have the N. And we can look at the variance there. And that variance was accounted for in the statistical models, okay? So the variance within herd and among countries, you know, all of those things were accounted for in the statistical models. Yeah. Well, okay, well, conclusions, and then I'll take those two questions. Is that okay? Okay, conclusions, we could categorise them. Acidosis risk differed. High-risk group had fermentation and bacterial changes associated with rapid fermentation. 6 ruminal phyla associated with acidosis risk, sugar and CP, vital predictors. Evidence of interactions with the mammalian genome, etc. And there may be markers for that ruminal environment. And overall conclusions, yes, it's a problem. Rumen pH only does part of the job. There are differences in types and causes of acidosis. Control is around the safe sequestration, and I would add removal of hydrogen. Amounts of forms of substrate are a major problem, or major opportunity is the best way to think about it. Understanding the ecosystem will allow us to improve. So I think Kurt and Gary, you guys had questions.
Sorry, what was it? I have 2 questions. Do you want the awkward question or the nice one?
You go nasty. I know you can be nasty.
No, the awkward one is, I was really delighted to see a lot of your information, including published stuff on variation between cows. Because when I've been working with them, I've had 2 cows, one in Wisconsin and one in New Zealand, where their rumen pH dropped down below 5 several times a day, and they were perfectly healthy.
Yep.
I mean, the silly one in New Zealand actually preferred bloody phytobeat and was eating about 94% of phytobeat. The one in Wisconsin, she was on a TMR, but her fibre digestion was very slow because the pH was so low. So my question is, how is it that some cows can tolerate this acidity in the rumen? Oh, and it was measured by me in the mid-rumen, in the top, okay? Because I shoved it in.
And that then means it's a profoundly low as opposed to the anterior reticulum. Yeah.
So how do they tolerate it? I mean, these are perfectly healthy. They weren't lame or anything.
Well, I guess the question is, do they? Because erosive lesions in the rumen, you know, they really do trigger some of the problems Greg was talking about. I think that you also have a huge degree, and that was what we were seeing with our study, where they just, you know, you look at it as differences in lactate, and some of them got virtually no lactate, and some have got 100 millimoles plus. So I think that variance, that was what triggered us to go into the genome. You know, like that amount of variation says to me that there's other factors on deck and we better understand them. I think that clearance rate, incredibly important. If they've got more ability to clear and if they've got better ability to sequester, and you know, we need to identify how efficient those animals are. As you say, the one up in Wisconsin may not have been working all that well, fibre digestion down, but the ones that can retain fibre digestion and deal with that rumen pH, you know, we want to know about those.
Okay, so my next question relates quite a lot to that.
Okay.
I used to work in bloat quite a few decades ago, but most of the good bloaters are dead now.
Yeah, absolutely.
And recently, this is a bit complicated. I stored away some samples in 1993 and someone found them at -80 and analysed the microflora in my best bloater and an ordinary cow.
Yep.
And it was completely different. And what was more interesting is my best bloater, I don't know if it was Moor or Curly Horn, one of the others, I think it was Moor, a prick of a cow. She digested stuff in nylon bags in the first 4 hours of digestion 80% faster than her compatriot who was normal.
Yeah.
So imagine that, 80% faster digestion in the nylon bags. And now I've actually got the microflora all analysed, but it just highlights that somehow these cows are regulating it, and I would like to know why. Do you have an answer? But don't waste your time.
No, I don't think I've got an answer. I think, look, what you're raising is the excitement to me. You know, these are fantastic observations, and I agree about the puffer cow. The puffer cow has much diminished presence. So, you know, again, like the laminitic cow, I think there's a much diminished presence. And, you know, those high rates of selection are an issue. But the opportunity, I think you're pointing out to me, and it's something I've pondered deeply too, is that if we think about the modern dairy cow, you know, the modern Kiwi cow should do, you know, 12,000, 14,000 litres, you know, on a genetic basis without too much trouble. So the question then becomes, you know, in another few years when you and I are buried, or whatever, there's going to be cows sitting at 20-odd thousand litres because those cows exist in that population. And we've got to identify that combination of mammal and microbiota that gives us those advantages and hopefully confers a health advantage as well as a production advantage on those critters. Am I allowed one more? Yeah. Okay, thanks.
Yeah, quick question because I know it's lunchtime.
Well,
you quite nicely showed this link between rumen microbiota and acidosis. Does that offer a big opportunity then for probiotics and even prebiotics?
Well, I'd have to say that probably the reason that we got supported in doing some of the research is that I suspect the company involved has more data even than we've got on exactly what's in there. So I think that, you know, we've got comprehensive data down to species level, but I think that's where people are going to be mining. But the only comment I'd make about that is that it is a brutal ecosystem. You know, the bovidae are so successful as a species around the planet, right? Because they've had this remarkable capacity to take organisms that are a benefit and accumulate them and put them in their rumen, right? So they're one of the big winners in the animal world. Now, it strikes me that very few of the interventions so far with probiosis have worked. And the reason is that it's a bloody brutal environment. You know, there's, what, 10 to the 23rd organisms in there. So there's many, many more bacteria in one rumen than humans on the planet. And when you start thinking about those things, you've got to think about who's surviving, who's winning, who's losing. And you just go and give them, you know, let's say you put in something fairly powerful, 10 to the 8th, you know, and you drop it in there, but it might be cleaned up in no time flat. So it's whether there's a capacity for that organism to make a difference. And I also would point out that when you're thinking about that, you know, the other thing to contemplate is this sort of black queen hypothesis and the biofilms. And, you know, you can have bacteria that don't even have a cell wall, and they're supported in a biofilm because they're pivotal to that metabolic pathway with two interdependent bacteria or a group of interdependent bacteria around them. So they can be a facilitator. So, you know, that whole rumen complexity, you know, it's just opening up now. It's going to be profound. So sorry to take a long time to answer, but yeah.
In the 1970s, there were a lot of deformation experiments and they never worked.
Absolutely. There was actually, I lived pretty close to the one that ICI was based at, and ICI, I forget how many chemicals they tried. It was like 1,100 different deformating agents, and guess what? Our mates, the protozoa, kept coming back. So, yeah.
Outstanding. Thank you very much, and a really fascinating talk. So, round of applause. Right, we're just going to swap the microphones over and we'll jump straight into Michael's talk. So I may as well introduce him while we're waiting.